My name's Michelle West and I'm a Professor of Tumour Virology here at the University of Sussex in the School of Life Sciences. Epstein Barr virus is a virus that infects white blood cells that circulate around in your blood, and they're the kind of cells that can turn cancerous and develop into a lymphoma. And Epstein Barr virus is associated with driving the formation of a lot of different cancers from these white blood cells, lymphomas, and that includes Burkitt lymphoma, Hodgkin's lymphoma, and also the lymphomas that arise in patients that have had transplants, so have a low immune system, for example, either a stem cell transplant or a solid organ transplant, or even patients that are suffering from AIDS, for example, where their immune system is low. The virus was first discovered actually because of its association with Burkitt lymphoma. So Burkitt lymphoma occurs at high levels in Africa, where it is particularly prevalent in children aged between about 6 and 8 years old. And it was first discovered in 1964 by a scientist working on cells from one of those Burkitt lymphomas, and he found that he could see a virus under the electron microscope in those cells, and he later went on to prove that it was a virus that could drive those cells to keep growing indefinitely, which is what cancer cells do. And that's what Epstein Barr virus does when it infects those white blood cells. It drives them to keep growing indefinitely when they would normally only survive for maybe 2 or 3 weeks. That's their normal lifespan. So that's how it drives them to become cancer cells. That's the confusing thing about Epstein Barr virus, is that across the world, more than 95% of us will be infected with the virus. Probably you and I are probably infected. And for most of us, we don't have any ill effects. We get infected when we're babies or small children. The normal route of infection is through saliva, so through kissing or from sharing cooking or eating utensils. We don't know we've been infected, we don't have any ill effects, we don't go on to develop Epstein Barr virus-driven cancers. And that's because we have a really good immune system, and our immune system recognises the cells infected by the virus and destroys them if too many of them grow. So the virus can hide in these white blood cells that it infects. It shuts itself down so that it's invisible to the immune system. It lives within us, we live happily, and we both coexist together. So certainly what you said is true. If you're then unfortunate enough to have something that affects your immune system, so you're given drugs to stop you rejecting a transplant, for example, or you have something that suppresses your immune system, you are then at risk from developing an Epstein Barr virus associated cancer because your immune system is gone, there isn't that check anymore, and the virus cells can grow out of control. So that's one way of getting an EBV associated cancer. But the other way has nothing to do with having a problem with your immune system. It has to do with other events that might happen in those cells that we don't fully understand yet. So random events that might happen to some people and not others. So for some Epstein Barr virus driven cancers, they think that there is other genetic events that happen, other changes in genes that are just random. There might be environmental factors. And for some of the cancers, we just don't know what the other factor is. So it's not always not having an immune system or having a problem with your immune system that causes the cancer. It can be another random event that, taken together with the virus, will push those cells down the cancer route and stop your body being able to cope with the numbers that you've got. So in terms of trying to combat Epstein Barr virus driven cancers, there's different ways you can do that. Because it's a virus, we've had a really big success recently with developing a vaccine against another cancer-causing virus, the human papilloma virus that causes cervical cancer. We now have a vaccination program for that virus that will prevent most cases of cervical cancer. So because you have a virus that's driving cancer development, you have the possibility of vaccinating against it. And that certainly is something that researchers are working on, trying to develop a vaccine that could prevent infection in the first place, so a preventative vaccine. That hasn't been successful yet. There have been quite a few problems developing a vaccine that works. And there are lots of researchers across the world who are still trying to figure out what they need to do and try to get a better vaccine. The other side of using a vaccine can be to use it as a therapy, which sounds a bit confusing. So you would normally think of a vaccine as stopping the infection in the first place, but you can develop a vaccine to use in patients that already have an Epstein Barr virus-driven cancer, to try and get their immune system to recognize the cancer cells. So if you vaccinate them and then you induce an immune response, those antibodies that the patient is producing will then go and attack the cancer cells. And there are early clinical trials that are being carried out using a vaccine for therapy. The other way to target those cancers is to develop new drugs that will target those cancer cells. And that's what my research really is more aimed towards the future of. We're trying to understand what happens in those virus-infected cells, how the virus changes those cells from normal white blood cells into cancerous white blood cells, so that we can figure out how that happens and then hopefully find a way to try and stop it or reverse it. We're hugely grateful to all the fundraising you and all the other people that raise money for Bloodwise do. It's hugely important to us, and we're really committed to carrying out this research. So what happens with science funding in the UK is that scientists like me need to apply for money to employ people to carry out the research in their labs. So we have jobs within research institutes or within universities where we may have some other roles in teaching, but our role at running a research group requires us to bring in money to fund the research. The universities or the institutes we work in generally don't give us the money to carry out the research. We have to go out and get that money. And there are various places we can apply to for funding. There are some government funded organisations. So for medical research, that would be the Medical Research Council. There are also a lot of cancer charities, so Cancer Research UK and Bloodwise being examples of those. And also charities like the Wellcome Trust, which fund a lot of medical research. So in order to get money from one of those organisations, we have to write a proposal, a grant proposal. And these organisations generally have certain dates a year where you are allowed to put in. They have a grant deadline that we would apply for money to. We write a proposal, we explain what research we want to do, how we want to do it, why it's important, where it could lead, and why we're the best people to do it. You then submit your application and it's sent out to experts in your area who will give it a score, write comments on it, decide whether they think it's good or not. And then a panel will meet of expert scientists at Bloodwise, and they will take all of those comments and those scores, and they will make the final decision out of all the people that applied for funding, which projects get given the money. So the money that you raise and you give to Bloodwise will be held in a fund, and that money will then be transferred to the universities or the research institutes that are successful in getting those projects funded. So for Bloodwise, the kinds of projects that they fund are for us to carry out research over a period of 3 years. And these are called project grants. And they are in the order of around £250,000. So a 3-year project grant normally adds up to a total of around £250,000. And roughly half of that will be the salary component of the person that we employ to carry out the research, so the expert scientists that will be working in the lab, including all of the salary contributions that the university has to make. The rest of the money we use to buy the equipment that you can see around you, so pipettes for carrying out experiments, centrifuges, water baths, other kinds of machines that we use, the facilities to grow the cells in culture, machines that carry out some of the more detailed experiments, and also the things that we need to use to be able to do the experiments day to day. So the gloves that we wear, the pipette tips that we use, these things are called consumables. They're things that get used up and that we have to replace. So science requires a lot of specialist equipment. It requires a lot of specially engineered individual pieces of consumables, flasks to grow the cells in, tubes to do the experiments in, all of those things. And all of that together would form the other half of the money. So roughly half on employing the expert scientists to do the work, and roughly half on the equipment and the consumables required to carry out the work. But the research in my group really concentrates on what Epstein Barr virus does when it gets into white blood cells, how it changes those cells. And we're particularly interested in how it switches on genes that make the cells grow, and how it switches off genes that might stop the cells from growing or enable the cells to die. And we discovered the way in which the virus switches on one of these key genes that forces the cells to grow, and it's a gene called MYC that is also switched on at high level in lots of other cancers, not just blood cancers. So lots of people are working on this gene. So the work we do has implications for lots of other cancers. And we found that the virus uses a very specific way to switch on the gene. And this differs to how it's switched on in other circumstances. But with relevance to Burkitt's, what we also discovered was something really interesting. So I told you that when Epstein Barr virus drives cancer, sometimes there are other genetic changes that happen as well that contribute. In Burkitt lymphoma, we know that one of the genetic changes that happens is a genetic change in this MYC gene. It gets a break in that gene, and that switches it on at high level. So you have Epstein Barr virus here, and you have the MYC gene being broken in a certain way that makes it switch on very fast. And what we never knew was whether Epstein Barr virus is doing things over here and this is happening here, and the two together are driving the cancer cells to grow, or if the virus is affecting this break in the MYC gene. Is it promoting it? Is it making it happen more often? So that's also how it's making the cancer develop. And what we knew was that in lymphomas, in Burkitt lymphomas that have the virus in them, they have a break in a specific part of the MYC gene that's different from in lymphomas where there isn't a virus involved. And the research we've done is giving us a clue that suggests that yes, the virus might be contributing to where that break in that gene happens. So that is telling us that yes, there may be this link. So the two things might not be completely independent. The virus might be doing something over here to the MYC gene that's making it have a break in a specific place. So it might be, contributing to the development of the cancer by switching on other growth genes and switching off death genes, and by maybe changing this MYC gene itself. And that's something that has been thought about for a while, but there's never been any evidence of it. And we have the first piece, just the first piece of the puzzle. We need to do more work. So we're going to be mapping more of these breaks in collaboration with some other Bloodwise funded researchers. So that's one side of the story. The other side of the story was working on a gene that is normally a gene that can induce death of cells. Okay? And this gene gets switched on and induces the death of cells when it senses that something's gone wrong in the cells. And those kind of genes are really important in our cells. Our cells need to grow, but they also need to know when to die, particularly if they sense something's gone wrong in them. And we know that Epstein Barr virus stops a lot of death genes like that being switched on because it doesn't want the cells that it's in to die. And we showed how it switches off a key death gene, a gene called BIM. And we also showed that we could use a drug to reverse it, switch the gene back on and make the cells die. And that's the drug that we were talking about earlier on. There's a set of drugs that target a protein in the cell which is called EZH2. Okay? And this is a protein that can be switched on at high levels in a number of lymphomas, so not necessarily ones that involve the virus. So this drug has been developed to try and target those lymphomas because they know that the fact that this protein is there in high levels in these lymphomas is driving them to grow. So the drug has been developed to stop that protein working and to kill the cancer cells. Still in early stages, but it is being looked at as a therapy for lymphoma already. And what our studies showed was that it might be possible to use this drug in lymphoma cells that are infected with Epstein Barr virus to try and induce the death of those cells, because the virus uses this protein to switch off this gene, this BIM gene. We need to do more work on that. We need to study more cell lines, and we need to figure out whether that's a viable option. And then it would move to looking at early stage trials of whether it could be included within existing therapies. So the point you made about drugs to be used when patients have relapsed or perhaps have not responded to treatment is a really important point, because it highlights something that is a problem in cancer treatment at the moment, which is resistance to drugs. So what can happen when a patient is treated with one drug or a combination of drugs? Those drugs could kill some of the cancer cells, but maybe not all of the cancer cells, because some of them survive because they have something within them that makes them resistant to those drugs. And those cancer cells, well, they'll be able to grow out, and that would lead to either a failure in treatment or maybe a relapse. So we're working a lot towards trying to develop new drugs so that we have another drug that we could go in with, to which those cancer cells would not be resistant. So a drug they haven't seen before, a drug that is targeting something different to the original therapies. And the more research we do to try and understand what we could target with these new drugs will lead to the development of more new drugs that can solve the problems with resistance to therapy or problems with relapse or failed therapies. Rest assured, there are lots of us across the world who are working on Epstein Barr virus lymphoma, and basic scientists like me trying to understand how the virus drives cancer development, people working on a vaccine, people working together with patients. And the way in which we publicise our research so that other scientists can benefit from it is through publications. So we publish our work in journals that are then available for people to read online or in print form. So we get our work out there like that, and that anybody can pick up on that research and carry on with it or contact me to collaborate. We also very much have a network of people that work on Epstein Barr virus. So we have an Epstein Barr virus association, the International Epstein Barr virus association, that I'm secretary of. And every 2 years we have a big international meeting where everybody presents their results and we all talk to one another. We have lots of collaborations within the Epstein Barr virus field. So that's internationally and nationally. And also within Bloodwise, we have an annual event every year called Grant Holders Day, where we go and selected people give talks on their research. I gave a talk a couple of years ago, and that gives us the opportunity to find out what other Bloodwise researchers are doing and to set up new collaborations. And as part of one of those meetings, I met up with another scientist who's working on particularly pediatric lymphoma, including Burkitt lymphoma, and we're hoping to be able to work together, particularly focusing on some of our latest research where we think the virus might be contributing to these genetic changes in this other gene, MYC. And she has some data that we might be able to ask some questions of, and she might be able to do some more analysis for us. So we are always finding new ways to work together and to collaborate, and the scientific community is very much like that.